Hi, thank you for having me. And I'm here to talk to you about a recent innovative approach for rare diseases that Vertex has experienced in cystic fibrosis. A little bit about Vertex. We are a young, mid-sized, Boston-based biotech company. We are focused on rare diseases. We have been able to serially innovate from bench to bedside and have brought 4+ drugs from bench to bedside in the last several years. Our primary focus has been on cystic fibrosis. In the last 5 years, we've brought 3 products to patients that actually treat the disease and not just the underlying symptoms. And I'll focus my presentation on, you can see the graphic there on our marketed products, development products, and out-licensed development. I'm going to focus today on Kalydeco and our journey that we've had with the FDA for Kalydeco and an innovative approach we've taken with the FDA to make treatment available for patients with rare mutations. Just a little bit of a backgrounder about cystic fibrosis. It's a rare, lethal disease affecting 75,000 people worldwide and 30,000 individuals in the United States. It's an autosomal recessive genetic disease. The mutations or errors in the cystic fibrosis transmembrane conductance regulator, or abbreviated CFTR, lead to absence or reduced amounts or inadequate function of the CFTR chloride channel. The result of this is that it causes mucus in various organs to become thick and sticky. It leads to organ issues like lung infections, respiratory failure, poor digestion, and problems in the reproductive system. There is a high burden of therapy with cystic fibrosis. Because this disease manifests in young children, I can't really say enough about the burden on their families and their care providers. Historically, most treatment has been focused on symptoms of a disease. The median age of death for this is 30. So let me talk a little bit about the genetics of cystic fibrosis. There are approximately 2,000 mutations that have been identified. Approximately 300 are known to result in cystic fibrosis or other pathological manifestations. There are 1,200, of those 2,000, there are 1,200 mutations carried by 5 or fewer people in the world. So you start to see the magnitude of this rare disease. The variability doesn't stop there. There are 3 different distinct biological problems that can present depending on the mutation for cystic fibrosis. The first is a defective CFTR function. This is resulting in a decreased chloride transport across the CFTR ion channel. The treatment for that's approved, that I'm going to speak more about today, is Kalydeco or ivacaftor. It's a small molecule known as a potentiator. It increases the channel opening probability to further improve the overall level of transport of chloride across the ion channel. The overall level of ivacaftor-mediated CFTR transport is dependent on the number of CFTR channels and their responsiveness to the treatment. The second biological problem that we can see with, depending on the mutation, is a defective CFTR protein trafficking and folding. The approved treatment for that is Orkambi, which is a combination of lumacaftor and ivacaftor, so it's both a corrector and a potentiator. And we also have Symkevi, which is also a combination of tezacaftor, ivacaftor, and ivacaftor. Under investigation, as we just heard from Dr. Bryan, is our gene editing and mRNA treatments for the defective protein synthesis. Our goal at Vertex is to treat the vast majority of cystic fibrosis patients. Currently, we have eligible 34,000 patients for the approved products, Kalydeco, Orkambi, and Symdeko. We are expecting EU approval the second half of 2018. We are pursuing label expansions based on age, so working down the age categories, and also increasing the number of mutations that we are able to treat. We have triple combination regimens in late-stage development, and we are pursuing the gene editing. So let me give you a little bit of history about Kalydeco for those of you that may be interested. Kalydeco was originally approved in January of 2012 for patients 6 and older carrying at least one copy of the G551D mutation. This is also known as a gating mutation. It affects the channel gate. In individuals who have this mutation, the chloride ion gate remains closed, does not open properly. This mutation was selected because of its responsiveness to the potentiator. It's the first medicine to be approved to treat the underlying cause of cystic fibrosis and not just the symptoms. In February of 2014, Kalydeco was approved for patients 6 and older carrying one of 8 additional mutations, and you see those listed there. It expands, so we started with 1,200 individuals in the United States. We increased it by 150 with the approval in February 2014, and then in December, another 500 patients had Kalydeco as an available treatment if they were 6 and older carrying the R117H mutation. So, the challenges of further expansion. What I just described to you, the initial approval and expansions were based on clinical trial data. There were adequate numbers of patients available for study. But as we started down this road, clinical trial-based approach became more and more difficult because the mutations were less and less common. There were fewer patients we could enroll in clinical trials in the traditional sense. We had several discussions with the FDA about possible alternate approaches, including N-of-1 studies, use of biomarkers such as sweat chloride, and combining different CFTR-based subpopulations with efficacy determined from pooled results. Each approach had its limitations, and the FDA suggested an in vitro cell-based data to predict clinical efficacy. After working with the FDA, very pleased to tell you that in May of 2017, Kalydeco was approved for 23 additional mutations. So we substantially increased the number of mutations that had Kalydeco available as a treatment. Last October, the FDA published in the Annals of the American Thoracic Society. They also had a CDER blog and a presentation at the North American CF Society, basically outlining this innovative in vitro approach that we employed with the agency. I'm going to further describe the approach, but suffice it to say that this was a patient-focused approach that really embodies the precision medicine principles to drug development. It is unique because it allowed expansion of an indication by using cell-based in vitro system to support efficacy in CF patients with mutations for which clinical data is not available and likely not feasible to obtain. In summarizing the approach that the FDA utilized in order to be able to provide approval based on in vitro data, they described these 4 pillars for evaluating in vitro data to expand an indication in the absence of clinical data. We heard from Dr. Woodcock earlier on the need for the totality of approach for rare diseases. I think this really provides support to that approach. The 4 pillars that you can see here, and they were reiterated in the recent targeted therapy guidance that I'll talk about here in just a bit, are comprehensive understanding of the CFTR mutations and channels, and assay findings consistent with existing clinical trials. For cystic fibrosis, we had 30 years of data on the CFTR structure and function, the clinical data on thousands of CF patients, and we had an approved product since 2012. We also had large efficacy and safety database with well-established benefit risk that's been in place for some time. We also had a solid understanding of the mechanism of action, and I think that we can't underscore that enough, that reasonably predicted clinical response. What I'm showing here is, apologies, on the other side we talk about the in vitro, that it was a robust validated assay and verification of data integrity and findings. Again, continued dialogue with the agency for acceptance of the using chamber in vitro electrophysiology measurement system, that it was standard, it was well-characterized method for evaluating ion fluctuation. And the data demonstrated that the mutant CFTR channels expressed in FRT cells were validated. They also were consistent with clinical efficacy data that we had. The important piece here is that the in vitro system allowed for the assessment of changes in CFTR-mediated chloride transport in response to ivacaftor, Kalydeco, in FRT cells expressing mutant CFTR channels. The shift in chloride transport in response to CFTR potentiator above a certain threshold, in this case it was 10% of normal, should have demonstrated that it's reasonably likely to identify mutations for which ivacaftor would provide a clinical benefit. That's really the key to this innovative approach. After this approval in May of 2017, the FDA came out with the targeted therapy guidance in December of 2017 and really provides recommendations building on this concept of an innovative approach and for rare diseases. The key points here is that it provided recommendations on grouping patients with different genetic mutations for eligibility in clinical trials, recommendations for evaluation of benefit risk. The agency will accept patient groupings if reasonable to expect grouped patients will have similar pharmacological responses. And last but not least, the agency generally will approve drug for all subsets included in the grouping, irrespective of whether subsets were represented in the clinical trials, which the Kalydeco example was a good demonstration of that. Lastly, because I realize I'm right up to the minute here, I want to briefly discuss the FDA building on this concept of accepting in vitro data for rare diseases in the recent, and as recent as last month, August of 2018, the approval of Amicus Galafold for Fabry's disease. This approval shows FDA building on the in vitro acceptance for a disease for which there were very limited treatment options in the last 15 years. Primarily, treatment consisted of enzyme replacement, and at least 50% have been untreated. The approval was based on the demonstration of 348 amenable mutations based solely on the in vitro data and very limited clinical trial data. The in vitro assay that you can see there was to identify responsive mutations on the basis of relative and absolute increases in the alpha-Gal A activity. And really, just to underscore that this is an innovative approach taken with the regulator for rare diseases that we've had the fortune with Kalydeco and look to the future for this approach. Any questions? I'm between coffee and... Great. Thank you.
The Amyloidosis Research Consortium was founded in 2015, and although there were a number of patient organizations, the rationale for founding the ARC was really an understanding that there had been a drug that was developed, safe, effective for a percent of the population, and did not gain approval. It's been approved in 40 countries outside of the US. And what came to light was really that there were significant barriers and challenges in drug development, in amyloidosis, and I think across rare diseases, and that there needed to be more work and structures put in place to help build collaborations to ensure that we have successful trials and break down those barriers to accelerate drug development, particularly for diseases that have no treatments. So I'm going to talk just a little bit about two types of amyloidosis. And these are the two most common types where treatments are in development. And so first of all, AL amyloidosis it's a misfolded protein disease. The abnormal protein is formed in abnormal plasma cells. And these are free light chains. And these light chains aggregate and deposit as fibrils, amyloid fibrils, in multiple organs in the body. of note, and I'll reference this a bit later, is the fact that particularly with AL amyloidosis, what we find is that those free light chains in themselves are toxic. So even without binding, can form as they're free light chains, can actually cause cardiac toxicity and damage. For ATTR amyloidosis, which is another type of misfolded protein disease, it is a hereditary variant, and there's also an age-related mutation that forms. The misfolded protein is actually a transporter molecule called the transports transthyretin, TTR, and that destabilizes and produces monomers that again aggregate, misfold, form amyloid fibrils, and form damage in the heart, GI tract, and peripheral nerves. So when we look at this, and I have only, first of all, I want to just preface this by saying that we have had a phenomenal working relationship with FDA. We are so appreciative of the time and the commitment that they've given to the amyloidosis diseases and working with us not just as a patient organization, but across all the sectors of the industry. So with the number of products we have in review, what I think is interesting to look at is that we have, for AL amyloidosis, we have 2 products that are in the hematology oncology division. And both of those have been able to be evaluated. Their primary endpoints are the hematologic response, i.e. the reduction of those free light chains, which then will imply that there would be less deposition and therefore damage. However, in the neurology division, where we have the hereditary ATTR, peripheral neuropathy patients are being, the endpoints, the clinical endpoints, which are the modified NIS +7, the MNIS +7, and Norfolk Quality of Life. The Norfolk Quality of Life is actually a diabetic neuropathy score, not one that's amyloid specific. And the modified neuropathy impairment score is a complex score which has challenges, as I'll talk about a bit further on, but these are clinical outcomes. However, the therapies that have reached the cardiovascular renal division, tafamidis for the cardiomyopathy patients, and also an AL drug, NeoD, have had all-cause mortality and frequency of hospitalizations as their endpoints. Whatever the rationale has been within the FDA, companies have really struggled and are trying to design trials that will in AL hit the hematology oncology division and in HATR hit the neurology division. So there are a number of concerns about that. And when we look at the phenotypes for ATTR, what we find is that patients present with neurologic or cardiac symptoms. But it's not that simple, because that might be the leading presentation. But in fact, these patients have a combination of cardiac and neurologic symptoms. And actually, the more we've started to understand, the finer our scales have got to evaluate these, we actually find that these are very complex, multi-systemic diseases. They're not one or the other. But what we're seeing that has been created is an artificial divide between this disease, creating hereditary ATTR polyneuropathy, hereditary ATTR cardiomyopathy. And what this really does, that unless you are one of the variants, which is really predominantly cardiac, like the V122I mutation, or the VALMET30, who have significantly predominant neuropathy or cardiovascular for the V122I, what this does is it just pushes the problem upstream. And now we are struggling with the fact that this treatments that are meeting approval will be approved for an incredibly small subset of the population. And what we haven't studied in these trials is the larger cohorts. We have, in some cases, had where they're studying the neuropathy, had some investigational endpoints for cardiomyopathy. But in other studies, we haven't. So we're actually pushing the problem upstream, creating immense risks for patients who have other organ involvement. And prescribers will be doing anything they can to find some neuropathy to prescribe these treatments. Amyloidosis are progressive fatal diseases. There are no other treatment options. So it really becomes critical about how we look and assess these. The other thing I would say, just as I mentioned previously, and I'll actually come back to, is the fact that, as you noticed with AL, we are able to use the biomarker, knockdown of free light chains. TTR, a similar process. Nobody has been able to use a knockdown of TTR, which would indicate a similar progression towards a disease or regression. So I want to talk a little bit more in detail about two other points and experiences that we're having. So just to look in a little more detail about AL amyloidosis, about 8,220 patients are diagnosed each year with AL across the US and Europe. 74% of those will have cardiac involvement. However, 40% of those patients will be too sick to benefit from any treatment and will die within 6 months of diagnosis. When we break down and think about what those numbers mean, when we look at the Mayo Clinic, the world's leading center for treatment of AL amyloidosis, in a period from 2000 to 2015, they saw exactly 2,553 patients with AL amyloidosis. So we have to think about, when clinical trials are designed, how we reach these patients and what the real numbers of patients that are accessible to trials are. One of the studies that has been referenced a number of times by the FDA as best practice is a study, and I won't go into great details about it, but it was a study in the general heart failure population. It took place in Scandinavia. It was called the Consensus Study, and was considered a landmark trial in severe congestive heart failure. This trial, when we look at the number, there were 253 patients that participated in the trial. Of 400,000 patients that were eligible, that only required 0.006% of patient participation. So 1 in 1,600 patients had to be enrolled in that trial. This trial was a huge success. It enrolled and actually completed within 8 months. So what would that look like for AL? With 1,500 patients eligible to participate, it requires 17% of the patient population. That means 1 in every 6 patients that's seen with cardiac amyloidosis would have to be enrolled in the trial. We actually recently completed our first prospective phase 3 multi-center trial with 110 patients, and it took 4 years and 10 months to enroll. Which brings me to my next point, the importance of biomarkers. And this is looking at that study is how many patients it would take and the length of time that it takes for us to do a clinical trial in AL amyloidosis. One that started in 2016, we'd get the last patient enrolled at the end of 2019, and results in at the end of 2021. However, if you model it on a biomarker, NT-proBNP, which indicates toxicity in the heart and damage in the heart, what we know from AL patients, it's not the knockdown of free light chains that's important, it's the cardiac damage that matters. If we used NT-proBNP as a biomarker, the difference would be that the trial, if it had enrolled in 2016, would be reading out right now. So this really talks about the critical need to understand, have flexibility, and be open to developing biomarkers to use as surrogate endpoints in clinical trials. Now, I want to talk for a moment about the Rare Disease Program. I know we have a number of people from the FDA who are part of the Rare Disease Program, and I would like, first of all, to say that we worked incredibly closely with Jonathan Goldsmith, who is the Associate Director of Rare Diseases in the Office of CEDAR. And without his support and help, our programs would be nowhere near where they are today. He was an exceptional advocate and thought leader amongst us in helping shape our programs. What I would say is, as we're looking at what we are developing to improve rare disease programs, the frustration that we've had is that while individuals have been very open to working and thinking about this, when companies have requested members from the rare disease program to attend their review meetings and their INDs, they've been told it was not necessary by individual divisions. So I think it really highlights that with these complex diseases, we need expertise. We cannot, I think we've been incredibly lucky to have had the commitment from FDA and time that they have given to amyloidosis. We have had an incredible number of meetings. They have come to all our research meetings, to our strategy meetings, to our scientific meetings, to our patient-focused drug development meetings. We can't expect that to be done across all rare diseases. And then we need to see that that effort that is being put in and invested in this disease is translated across divisions. Because in the end, the cost it is to patients when drugs are approved and they don't have access and there isn't reimbursement is really comes back to this very need to think about how we're working and developing from this get-go. Thank you.
Time for one question. Does anyone have a question? All right, thank you. No, thank you. Welcome.
Annie Kennedy from Parent Project Muscular Dystrophy.